Hello students. In the previous session, we had learnt about two experiments. One was called as transformation principle experiment, explained by Frederick Griffith in the year 1928. The other set of experiments was called as biochemical characterization of the transforming principle, that was given by three people, Avery, MacLeod and McCarthy. You need to remember the name of the scientist along with the experiment. Right? See, in the biochemical characterization of the transforming principle experiment, they had proved that DNA itself is the genetic material which is responsible for the transformation of the R strain into S strain. Yes? So this was confirmed chemically, right, or biochemically. The molecular structure or the molecular study of the DNA had already started, and it was confirmed that it is the DNA itself is the genetic material. But it is science. The scientific community always disagrees with one or the other arguments, right? People of science, they do have a tendency to argue for everything. It may be right or wrong. The simple or something like that happened here also with the MacLeod, McCarthy and Avery. Some of the people, they disagreed with them. They did not agree to the proof that was given by these three people. And then in the year 1952, another two people, they conducted another set of experiments to give an unequivocal proof to the fact that DNA itself is genetic material. Okay? DNA as genetic material, or DNA itself is genetic material. Okay, see, this experiment is known as the Hershey and Chase experiment. Hershey and Chase experiment, or it's also known as blending experiment. Okay, or it's also called as radio labeled blending experiment, also in some books they refer it. So it was called as Hershey and Chase experiment because the two scientists who did this, who performed the experiment in the 1952 are Hershey and Chase. Alfred Hershey and Martha Chase. In the year 1952, they gave this experimental proof to say, or to give an unequivocal proof. Unequivocal proof for DNA is genetic material. So this was confirmed firmly by this particular experiment. So what did they do? See, before going to understand the experiment they conducted, we need to know what all materials or what all the tools that they used here. See, they considered bacteriophage, T2 bacteriophage. Bacteriophage was considered. You know already bacteriophage means it is nothing but it is the virus infecting upon the bacteria. They considered the E. coli cells, you know E. coli also, Escherichia coli, the bacteria, right? So, and they considered radioactive sulfur, that is S35, see, remember, and also radioactive phosphorus, P32. Okay, radioactive sulfur and radioactive phosphorus were considered. So, why did they consider? What is the basic idea behind this experiment? See, they knew that. See, you know already, this is the bacteriophage. The diagram of the bacteriophage is commonly drawn like this. It will be having a protein head which is called as the capsule, and it will be having the neck region and the tail fibers. So already know the structure of the bacteriophage. And you know the bacteriophage will be consisting of the genetic material, and that genetic material will be what? It will be the double-stranded DNA. Yes, it will be always what? Double-stranded DNA molecule. Yes, circular DNA molecule. Now, protein coat is there and DNA is there. What is their job now? With this experiment, they just want to prove that whether the protein is genetic material or DNA is genetic material. As I told you, by the year 1928, 1930, when Frederick Griffith he completed his experiment, so many of them they believed that protein is genetic material. Even after the biochemical characterization of the transforming principle experiment also, they were not ready to agree that DNA is genetic material. Some of them, they still believed protein is genetic material. And with this experiment, it is crystal clear that it is not protein, it is DNA is genetic material. How did they do? See, just to prove that, he had considered T2 bacteriophage. Okay, by understanding the structure of this, you should know this is nothing but the protein coat, or also called as capsid. You know that. It is also called as capsid or protein coat. So, they have considered radioactive sulfur. Sulfur is predominantly present in, it is present in protein. You know the structure of the protein, you have studied already in the biomolecules chapter. There are many disulfide bonds, right, which make the protein molecule, and the phosphorus is predominantly present in DNA. So you know phosphoester bond, phosphodiester bonds, right, and the phosphate group present at the 5th carbon atom of each nucleotide. So that means phosphorus is the major component of the DNA and sulfur is the major component of the protein. And sulfur is not present in DNA, phosphorus is not present in the protein. Phosphorus is present only in DNA, sulfur is present only in the protein. So this nature of these two molecules, they took the advantage of the nature of these molecules having sulfur and phosphorus respectively. Okay, they had radio labeled sulfur and phosphorus. Okay, you know what is radio labeling? Radio labeling means we are going to radioactivate the molecules, the phosphorus and sulfur, just to detect their presence in the cell or in the body. Okay, their presence can be detected because of the radioactivity, okay, of these particular molecules. Now with this idea, what did they do? See, they took two different cultures. On one culture they grew bacteriophages. See, this is one culture with radioactive sulfur, that is S35. I am going to write with this one, saffron color. In the other culture, the phosphorus supplement was radioactive and that was P32. I am going to write with the yellow, S35 and P32. The bacteriophages, they were grown on this medium. They had developed a radioactive protein coat. See, the protein coat was radioactive here. Yes or no? Because sulfur is predominantly present in the protein, and the radio, that means which part of this one has got the protein here? The part of the bacteriophage which is made up of the protein is nothing but the capsid. Yes or no? So that is why capsid is radio labeled, rest other portions are not radio labeled, right? That is why only capsid I have drawn with the saffron color. Whereas in this case, on this culture, whatever these were bacteriophages were cultured on this culture, they developed radioactivity with respect to DNA, not with the protein. Okay, only DNA was made radioactive because phosphorus is predominantly present in DNA. Okay, that is why DNA was made to shine or radioactive. Now these were made to infect. Okay, these batch of viruses were made to infect upon one batch of what? Bacteria considered in a Petri plate. Okay, so these cultures of bacteria were infected with the viruses which were having the radioactive sulfur, and this batch of bacteria were made to infect with the viruses which were having radioactive phosphorus. Now you know when infection occurs, the transferring of genetic material takes place. Okay, how? Not the entire body of the entire body of the virus will not enter. It makes a small pore here and it will transfer only the genetic material. Only the genetic material is transferred from the capsid into the body of the bacterial cell, right? Similarly, here also, what happened? See, it was consisting of the DNA, which is the radioactive DNA. So that was transferred to the bacterial cell, right? And this was done, see, this was the radioactive DNA. This DNA was not radioactive because transferring can take place only with respect to the DNA, not with respect to the capsid. Only the genetic material is transferred, right? So obviously this bacterial cell will not develop any radioactivity, whereas this bacterial cell will develop radioactivity because the DNA has been made radioactive there. Now what did they do? They cultured, they allowed them to multiply. Okay, multiplication happened. That means they allowed them to multiply or reproduce for 1 or 2 generations, and then what did they do? They centrifuged. They subjected it for centrifugation. What is centrifugation? Centrifugation means rotating at very high RPM. See, centrifuge means it's an instrument. See, there will be a stand and there will be 2 test tube holders here. Here you can fix 1 test tube, here you can fix 1 test tube. This is a mechanical one. In the machines, you can put more than 5 test tubes also, right? So there will be a handle and both will be having equal distance, and you will have to rotate the handle and it will rotate at a very faster speed. Okay, when it rotates, see, these test tubes are only held at this position, only at this position they are held. So the lower part is free. Now, when you rotate it at a very fast speed, like around 5,000 or 10,000 RPM, so then what happens? These test tubes, they will fly in the air like this. And because of this high rate of rotation, what happens? See, because of the centrifugation or centrifugal force which is acting upon that, see, the centrifugal force is continuously acting upon it. Now the lighter particles will come up. The lighter particles will come up and heavier particles will settle down. Yes or no? So the heavier particles settled down. Usually they are called as pellets. The heavier particles which are settled down after centrifugation, they are called as pellets, and the light fluid which will come up, that is called as supernatant. That is called as supernatant. Okay, now in this case, what happened here when they did the centrifugation? So neither the pellets nor the supernatant showed the radioactivity, whereas in this case bacterial cells were radioactive and they were heavy particles. They settled down to form the pellets, and that is why pellets showed radioactivity. Pellets showed radioactivity, whereas here no radioactivity. No radioactivity because phosphorus was labeled here, and that phosphorus containing DNA was transferred from the virus to the bacterial cell, and that is why bacteria got radioactivity, whereas here it was the protein coat, so protein is not transferred, that is why radioactivity is not seen. So by this it is once again confirmed that it is not the protein, it is the DNA which is responsible for transformation, which is responsible for inheritance, and DNA itself is the genetic material. It was proved unequivocally. There was no argument to this experiment once again, right? So this is also one of the important experiments which can be asked for 5 marks. I've told you it can be called as blending experiment or radio labeling experiment, or is also called as Hershey and Chase experiment. Or they also ask you like explain the experiment which gave an unequivocal proof that DNA itself is genetic material. Which experiment is that? Same experiment. Okay, so this is about today's session. In the next session we will consider the next aspects.